Hello, so we are going to talk about the gram staining. So this lecture will divide into two parts. One is Isme preparation and then the steps in gram staining. First, how to make a smear for gram staining. So we need a clean glass slide, right? Take a loop full of sterile water or normal saline. Put it on the center of the glass slide, right? It has to be a clean glass slide. Then take a small amount of bacteria colony from the culture plate, right? If you have a suspension, bacteria suspension, or a clinical sample such as CSF or pus, then you don't need a drop of water. You can use the loop full of the sample or bacteria suspension, put it in the center of the clean glass slide. Right? After putting it on the glass slide, you have to make the smear by spreading it out, right? So you move you circle the wire loop, right? Circle the wire loop so that a size of a dime is spread out, right? The size of the smear has to be about the size of a dime. But in case if you feel the smear is too thick, right, you can still spread out little bigger. Doesn't matter. You can spread it out bigger. The bottom line is it has to be visible and it has to be thin. Right. So after making the smear, right, next step is fixation. Right. So why do we do the fixation? Fixation means fixation of the smear to the glass slide. Now because during the gram stain and we will be using lot of stains, and after each step of using stain, then we will have to wash the slide with flowing water, tap water. So if you don't fix it, the smear will wash away. So to prevent washing out of the smear, right, we have to use the fixation. There are two methods of fixation. You can use air drying for fixation or you can use heat fixation. Right? Air fixation is, in the air fixation what you do is you just let the glass slide to dry in the air. The smear, you keep the glass slide with the smear on top of the on the upper surface, you keep it on the surface and let it air dry. That is air fixation. And heat fixation means you pass the glass slide to a Bunsen flame. You pass 2 or 3 times very quickly, right? And if it is little warm, that means it should be, the smear will be fixed with little warmth, right? So the fixation will fix the smear to the glass slide. It won't allow it to be washed away during the washing process or staining process. Right. At the same time, fixation also kills bacteria, so it makes the procedure more safer for you. But but still there may be live bacteria, so you will have to wear gloves when you do the gram staining. Wearing gloves is important. So when you talk about the steps in gram staining, right? So when you talk about the gram staining, is the most important staining method in bacteriology, and it was invented by Dr. Christian Graham. So that's why we call it gram stain, right? It's a very important diagnostic identification of bacteria in clinical specimens. It's very useful in clinical medicine because sometimes gram staining alone is enough to start the treatment. Right? In most cases, you can come to a most likely diagnosis or presumptive diagnosis like this. Now, take the first example. This one, right? You can see lanceolate shaped gram-positive diplococci there. If you see a sample, if you see in the gram chain, if you see lanceolate shaped gram-positive diplococci, right, is most likely pneumococci. So you can see pneumococci in sputum sample, a patient with pneumococcal pneumonia, or CSF sample, a patient with pneumococcal meningitis. If you do the gram stain and you find pneumococci, then you can call the ward physician and tell it looks like it is pneumococci. So you can start treatment for pneumococci. But confirmation of the pneumococcal is after the bacterial culture. But the thing is, bacterial culture take about 48 hours, and bacterial culture and antibiotic sensitivity. But with the gram stain, you give the report on the same day, though you don't have the confirmation or you don't have the antibiotic sensitivity testing. So with that gram staining, they can start the empirical antibiotic, right? That is most likely antibiotic that will be effective, right? Then similarly, this one, gram positive cocaine chains. When you see the gram positive cocaine chains, is most likely streptococci. So suppose you get a sample of pus and you see the gram positive cocaine chains. Before you don't, the physician don't have to wait for the culture report, right? We take 48 hours, right? We still send the culture report because that includes antibiotic sensitivity testing. But we can give the presumptive diagnosis on the same day. You can call them and tell it looks like streptococci. You can start treatment for streptococci, which is the drug of choice for streptococci is penicillin. Similarly, if you see gram positive cocaine clusters, that means it is staphylococci, right? So you can tell the ward physician is most likely staphylococci. You can start an anti-staphylococcal antibiotic such as nafcillin, cloxacillin, dicloxacillin, right? If it is a antibiotic, depend on the antibiotic sensitivity test, but they can start, for now, they can start an empirical antibiotic, right? Similarly, if they see gram-positive bacilli in the CSF of a neonate who has presented with the signs and symptoms of neonatal sepsis or neonatal meningitis, then it is most likely, if it is from CSF gram-positive bacilli, it is most likely Listeria monocytogenes. Then you can suggest to start treatment till the confirmation is given. They can start an antibiotic that cover Listeria monocytogenes, which is ampicillin. Ampicillin is the drug of choice for Listeria monocytogenes, right? So empirical antibiotic combination will be ampicillin plus cefotoxin or ampicillin plus gentamicin to cover other possible bacteria, but ampicillin will definitely cover Listeria monocytogenes, right? Similarly, you take urethral sample from a patient who comes with urethral discharge after sexual activity, right? Urethral discharge, and you do the gram stain. In the gram stain, you see gram-negative bean-shaped diplococci. Gram-negative bean-shaped diplococci. Some of the diplococci are within the neutrophils or pus cells. So that is diagnostic of, though you even without culture, diagnosis is confirmed. That is gonococcal urethritis. If it is from the cervix of a female, it is gonococcal cervicitis. You can suggest, you can send the report. Diagnosis is gonorrhea, gonococcal urethritis, or gonococcal cervicitis, right? Then the physician can start it, but most likely ceftriaxone 250 milligram intramuscular, just single dose, right? So you understand the value of gram stain, right? It is helpful in diagnosis too, in addition to the usual identification. So in gram stain, we distinguish two types of bacteria: gram-positive bacteria, which stains dark purple, and gram-negative bacteria, which stains light red, right? So in the gram staining procedure, we use 4 reagents in the proper order, right? There are several modifications of the original gram stain, but we commonly use the method that I'm going to explain, and that is, we use primary stain, mordant, decolorizing agent, and the counter stain, right? The primary stain that we use is a violet dye, aniline violet dye. It can be crystal violet or methyl violet or gentian violet. Gentian violet is a mixture of crystal violet and methyl violet. So primary stain, that is the primary stain that we use in gram stain. Then we use the mordant. After using primary stain, we use the mordant. Mordant is an aqueous solution of iodine. What is the function of mordant? It intensifies the color of the primary stain. It increases the color of the primary stain, right? The mordant. Usually we use Lugol's iodine. Then decolorizer. What is the decolorizer that we use in gram stain? Acetone alcohol. Acetone alcohol, very important step. Decolorization step. Then after decolorization step, right? Counter stain. We use the counter. Anything that will decolorize will be stained by the counter stain, right? We use basic fuchsin, neutral red, or safranin, right? All these steps in this gram stain, we commonly use all the stains should be there for 45 seconds to 1 minute. So after each step, after 45 seconds to 1 minute, we have to wash the slide with the running water, running tap water, right? So at the end of the staining, we have to air dry and do the microscopy, right? So we talk about this, and yeah. So when you talk about the steps in gram stain in detail, but I have another lecture, different lecture about the mechanism of gram stain, right? So first, this is little, we'll talk about it in a different lecture, but to have an idea about what happened during gram stain. So step 1, what did we do in this step 1? We added the violet dye, which is crystal violet, methyl violet, or Jensen violet. So what happened when you add the dye? Stain enters to the cell wall of the bacteria and it stains the organisms purple. Any organism, gram positive or gram negative, all will be stained purple with primary stain, which is violet dye. After that, after 45 seconds to 1 minute, you wash it with running water and then add the mordant, which is iodine. That is step 2, right? When you add the iodine also enters the bacterial cell through the cell wall, and then it forms a diiodine complex, crystal violet iodine complex, right? So the color of the diiodine complex is darker now because iodine intensifies the color. It mordant intensifies the activity of the primary stain. So the color becomes dark purple, right? So after step 2, both gram positive and gram negative bacteria will be dark purple. Then step 3 is decolorization, right? So decolorization step, we add the acetone or alcohol, right? So it will decolorize within 45 seconds to 1 minute. It will decolorize only gram negative bacteria, right? Gram positive bacteria will not be decolorized within 45 seconds to 1 minute because I will explain in the next lecture about the mechanism. Because the gram positive bacteria have a thicker peptidoglycan layer. So within 45, the timing is not enough for the decolorization to penetrate the bacterial cell wall and enter the bacteria, right? So the gram positive will not be decolorized within that time period, but it will easily decolorize gram negative bacteria because it has thin peptidoglycan cell layer, peptidoglycan wall, right? So gram negative will be decolorized. So that means gram positive bacteria will be purple, dark purple. Gram negative bacteria will be colorless now after step 3. Now step 4 and at the counter stain, right? Counter stain, right? Basifucrine, safranine, right? That is counter stain, right? Or neutral red. So it will list now when you at the counter stain, it will stain only gram negative bacteria because they are the only bacteria that are colorless. Gram positive are already dark purple. So gram negative bacteria will be stained pink with the counter stain because that's a pink dye. Counter stain is a pink dye. So the now gram positive, at the end of the gram staining, gram positive bacteria will be dark purple. Gram negative bacteria will be pink or light red, right? So that is about the gram stain. And you can see the gram negative bacilli here, gram negative bacilli, and these also gram negative bacilli, right? And this gram positive cocci. This is a gram positive cocci chain. You can see what kind. They are most likely streptococci, right? This is gram negative bacilli, right? They are most likely bacillus species. Gram positive bacilli. So they are most likely bacillus species, right? So that is about the gram stain. Hope you enjoyed the lecture. Thank you. If you found this video helpful, please like this video and subscribe to my channel. You can get new video notifications by clicking the bell icon. My Patreon supporters can request lectures and get exclusive content. Please check the description for links and more details. Thank you.